Welcome you all. I am T. Vinodini, lecturer, Department of Biotechnology from Sita Lakshmi Ramaswamy College at Tiruchirappalli. Today we see the session about the protoplast isolation methods and its application. First of all, the definition of protoplast. The protoplast are naked plant cells without the cell wall, but they possess plasma membrane and all cellular components. Why? The protoplast production of clones of plants that they produce particularly good flowers, fruits, and or have other desirable products. They quickly produce mature plants. The production of multiple plants in the absence of seeds, the necessary pollinators to produce seed. The regenerate of whole plants from the cells that have been genetically modified. The productions and the plants or containers reduces the disease and transmissions. Next, the sources of protoplast. The protoplast can be isolated from wide variety of tissues and organs that include leaves, roots, shoot apices, and the embryos and microspores. Among the leaves, especially mucophils tissues are mostly used. Callus and suspension also as a source of plants. Callus mean undifferentiated group of cells. Next, the types of isolation. The types of isolation can be isolated by two techniques, namely, one is mechanical method, another one is a enzymatical method. The mechanical method, it's a in vitro condition laboratory, is a crude and tedious procedure. In this result in the isolation of very small number of protoplasts. These cells are subjected of plasmolysis using hypertonic solutions. Then the tissue dissected to release the protoplast. Next, see there the slide. First of all, we isolated the young plant tissue and to examine under the microscopic the protoplast epidermis peeled out with the help of knife. Afterwards, the hypotonic solution help of the cell wall digested the protoplast, having digested the cell wall and taken the viability test. Then the protoplast isolation, intact leaves, a partial epidermis peeled off under dim room light. Then the stripped leaves in pre-plasmolysis medium in 15 minutes. To transfer to digestion medium under light intensity after 300 millimolar. Then the protoplast collected into washing media at maintenance pH 6. To centrifuge at 3 minutes, the protoplast pellet suspended in washing the media. Again, the centrifuge at 3 minutes, the protoplast pellet suspended and washing media at maintenance pH 7. Then the centrifuge at again 10,000 rpm 2 minutes, the protoplast pellet final suspension in the media using Morasuj and Skoog media. Afterwards, finally, the protoplast preparation stored in ice cold water. Then, second method, the enzymatic method. This method is widely used technique isolation of protoplast. The mechanism. The enzyme can digest the cell wall, but the protoplast only having plasma membrane and other cell organelles in it, but only digested cell wall only for record this method. Next, here the chemically the plant cell wall made up of cellulase, hemicellulase and pectin, so it was defeated by a respective enzyme like pectinase, etc. Then the slide, the shoot culture is taken about remove the leaves. After, cut the leaves and to incubate the enzymes using macrozyme, onasuka, and after incubate 12 hours. After 12 hours, 2% of sucrose solution used and isolate the protoplast. During this time, the enzymes are digested the cell wall. Then, using centrifuge at 10,000 rpm, 3 minutes, the pellet was collected. This pellet was collected and digest the cell wall protoplast taken to ready to for culture. Then, two-step sequential method. The enzymes are used one by one, first treated with pectinase and then treated with cellulase. One step or simultaneous method is both enzymes are simultaneously like pectinase and cellulase. Here, the young plant was taken using surface sterilization, leaf sterilization, then peeled out the epidermis peeling. The peeled out leaf segment was taken about and plasmolized the cell using hypertonic solution. The cells in enzymatic method mixture the cell wall and cell wall digestion and release the protoplast. After release the protoplast, maintenance the centrifuge and aqueous discarded the aqueous and collect the pellet. The collect the pellets also washing in protoplast. The protoplast are isolated the protoplast and plating of protoplast before selection and viability test. Afterwards, the cell wall regeneration forming the clump of cells and callus tissue. The callus tissue is nothing but a undifferentiated group of cells. These group of cells undergoing to differentiation of callus tissue. These callus tissue to derived from plantlets. These plantlet afterwards hardening and produce a young plantlet. Then, after the isolation, the protoplast, the following steps are taken: the purification of protoplast, the viability of protoplast, and culture of protoplast. Next, the application of protoplast. The application of protoplast, which will be involved in hybrid development. It means to develop somatic hybridization, whatever our interest plant. Then to regenerate the whole plant. Then they used to study the cell wall synthesis and composition. Then the study of cell fusion and somatic mutations, mostly used in genetic information passed throughout. Thank you.